So today we're going to be doing a lab with proteins. We're going to use a reagent called Burnett solution. Burnett solution will adhere to the polymer which makes a protein. Remember, proteins are made of 20 amino acids within the human body. We have what we call a carboxyl group hooked to the outside. This is one bond of the amino acid hooked to another bond of the amino acid. Right here we have 6 samples. Sample 1 is going to be water, 2 is egg, 3 is our powdered egg, 4 is powdered milk, 5 is going to be grapes, and 6 is going to be tomato. What we're going to do is we're wearing goggles with this lab because your eye fluid has polymers in it, but we're going to take 2 milliliters and mix that with our solution. After we've mixed it, we're going to invert it, which means to seal it and turn it upside down to see our mixture for this. It takes about 2 minutes for the mixtures to completely turn the color, while some will turn instantaneously. You'll notice as we're doing this, we're getting a nice color change with some, while others have no color change, or the color change is mild for this. What we're looking for is a protein to see if there is present or not present. Unlike our glucose test, we do not have to heat this, but it's an instantaneous test to test for proteins. In our sample 1 of water, this is completely negative. It is a blue color. In our sample 2, which is egg, you're going to notice that we have a yellow with a blue on top. As we invert this or mix this together, your egg will separate into 2. The yellow will stay yellow, while the blue at the bottom will actually separate and turn almost a purple color. You'll notice the egg is very chunky now. It's almost scrambled. And the purple is going throughout. What we see here is the yolk is what we call a zygote. That's the embryo. That will not turn a protein color because it does not contain protein. However, the egg whites do contain protein and most of the nutrition for the embryo. In our next test, or number 3, we did powdered egg. We can see it's a nice positive purple color. In our third, we did milk. Milk also turned a nice positive purple for proteins. However, in 5 and in 6, tomatoes here and grapes here, we do not see a color change. They're still yellowish in color. Part of the color change is just color theory, where things will turn colors based on adding 2 different colors together, such as red and blue would make purple. However, with this, it's not a positive test. These are also containing zygotes, or the embryonic form called a seed for these plants. They're surrounded by what the biomolecule that plants make in abundance, which is carbohydrates. So you will not see a positive protein test for plants, but if we did sugar tests on 5 and 6, we would see them turn a bright reddish orange, which we know from our carbohydrate test tells us that they're full of monosaccharides, or simple sugars. These 5 and 6 would also turn negative for starches because they're full of monosaccharides. This is the zygote. They're trying to feed their babies, and so they put a simple sugar that can be digested. Please make sure that you look at your protein tests, which were the positives, which were our negatives, and make sure you can give both qualitative and quantitative data for that. Remember, qualitative data has to do with your senses. What does it look like? What does it smell like? Now, obviously, you can't smell or taste things, but you can look and explain the color. While we have our quantitative testing, which tells us numeric values, make sure that you can tell us a numeric value for these, such as 3 out of 6 were positive, while 3 out of 6 were negative tests. And that is your protein test. Please make sure you fill out your guide and turn it in.